welcome to the Deep Dive. Today, we're tackling a topic that is, quite literally, reshaping the future of medicine. We're going deep on nanomedicine.
And we're using the book Nanomedicine, From Science to Business, as our guide. Our goal today is to give you a really complete picture.
A full journey, right? We're starting with the absolute basics, the quantum science, and we're going to build all the way up to clinical applications and even the business side of things.
Exactly. From the impossibly small to the multi-billion-dollar industries it's creating.
So let's start with that scale. When we say nano, what are we actually talking about?
Well, the word nano is from the Greek nanos for dwarf. And quantitatively, it means 1/billionth, so 10 to the minus 9 meters.
It's hard to even visualize that.
It is. You're operating at the level of individual molecules, of atoms. But what's fascinating is that this isn't entirely new.
Boy.
It's just the control that's new. We found that 9th century Mesopotamian artisans were using silver and copper nanoparticles to create this beautiful luster effect on their pottery.
No way. So they were nanotechnologists and didn't even know it.
In a way, yes. Today, the scope it's just massive. It pulls in everything. DNA sequencing, stem cells, regenerative medicine, computer science. You can't make progress without physics, chemistry, and biology all talking to each other.
Okay, so if we're at the molecular scale, we have to start with the real fundamentals, quantum basics. We're not talking about electrons as little planets orbiting a nucleus anymore, are we?
Not at all. That whole model is out. You have to think in terms of quantum orbitals.
Which are more like fuzzy clouds of probability.
That's a great way to put it. They're these complex shapes where an electron is likely to be, and the whole game really is about stability. Elements like neon or argon, the rare gases, their electron shells are completely full.
So they're stable, they don't react.
They're inert. But elements with partially filled shells, they are desperate to interact, to gain or lose electrons to get that stability. And that's chemistry.
Which brings us to the elements that matter for life. Hydrogen seems simple, but it's key.
It's incredibly versatile. I mean, it accounts for over 75% of all ordinary matter. It can donate its electron, acting like an alkali metal, or it can accept one, like a halogen.
But the big deal is hydrogen bonds.
The big deal is hydrogen bonds. These weak attractions are why water behaves the way it does. Water is a polar molecule. It has an electrical dipole moment.
Meaning one end is slightly positive, the other slightly negative.
Right. And this leads to cohesion water molecules sticking together and adhesion, them sticking to other things. It's why capillary action works in plants, and it's fundamental to how things move around in our own bodies.
You see that kind of duality in other elements too, like nitrogen.
Nitrogen is a perfect example. As a gas in the atmosphere, it's pretty inert, but its compounds they're everything. It's the basis for explosives like nitroglycerin.
Which we also use as a medicine to widen arteries.
Exactly. And on the other hand, you take a simple cyanide ion, a carbon, and a nitrogen with a triple bond. And it's one of the most lethal poisons because it shuts down cellular respiration. Tiny change, massive biological effect.
So from atoms, we build up. Let's talk about the macromolecules of life. Carbohydrates.
Right, the saccharides. Their main job, is accessible energy storage.
And then lipids. Fats.
Lipids have two big roles. You have triglycerides, your fats and oils, which are for long-term energy storage. But structurally, the phospholipids are non-negotiable.
Because they form our cell membranes.
They form the lipid bilayer of every single cell, that selective barrier. And if you're designing a nanomedicine, you absolutely have to understand how to get through it or interact with it.
And finally, the workhorses of the cell. Proteins, built from amino acids.
Yes, and the basic structure is always the same. An amine group, a carboxyl group, and a side chain, the R group, all attached to a central carbon. Only 22 of them are used to build proteins.
The critical part is how they link together, right? The peptide bond.
That's the key. Because of something called resonance, that peptide bond can't really rotate freely. It's restricted. And that limitation forces a predictable planar arrangement.
So it's like a flat, rigid link in the chain.
Exactly. And that initial predictable geometry is what dictates the entire complex three-dimensional folding of the final protein. If that geometry is off, the protein doesn't work, the drug has no target.
That structural control is really the foundation for all of nanomedicine.
It is. If you want to create new therapies, you have to be able to read and manipulate these structures. And for that, you need an incredible amount of data and computer power.
Let's talk about that data. The central dogma, as Francis Crick put it in 1958, RNA makes protein. The human genome is just huge. 3 billion base pairs.
It is. And what's interesting is that our understanding has refined. We used to think there were many more protein-coding genes. That number's actually dropped below 20,000.
Why is that?
We've just learned more. We realize that many regions are for regulation or they make non-coding RNA. The complexity is still there. We've just reclassified it.
And the technology to read all this information has evolved at a dizzying pace. Where did sequencing start?
The first generation was Sanger sequencing. It used a chain termination method with fluorescent dyes. It was good. It gave you long, accurate reads of maybe 800 to 1,000 base pairs.
For 3 billion base pairs.
It was incredibly slow and expensive. You'd have to stitch together millions of these long fragments. A monumental task.
Which is why we needed to shift to next-generation sequencing, NGS.
Right. Companies like 454 Life Sciences and Illumina changed the strategy. They said, let's prioritize raw, speed-high throughput over initial accuracy.
So you get more errors, but it's way faster.
Exactly. To compensate for the errors, you just sequence the whole genome over and over again, maybe 5, maybe 30 times coverage, and statistically you weed out the mistakes.
And that turned into a business challenge, the race for the $1,000 genome.
That was the benchmark. Bringing the costs down from millions to under $1,000 was what turned genomics from just a research tool into a plausible clinical tool you could use for diagnostics.
Now we're at the 4th generation, nanopore sequencing. This sounds like science fiction.
It's amazing technology. Oxford Nanopore is the big name here. They use a tiny protein pore, just a few nanometers wide.
Just big enough for a single strand of DNA.
Just big enough. As the single strand of DNA or RNA is pulled through this pore, each base, A, T, C, or G, creates a unique disruption in an ionic current flowing across the membrane.
And you can measure that disruption.
You measure it in real time, and that electrical signature tells you which base just went through. It's direct, real-time, single-molecule sequencing.
This level of precision, both in sequencing and imaging, really depends on computing. Let's talk about imaging.
Well, take the scanning tunneling microscope, the STM. It has near-atomic resolution, but it doesn't see with light. It feels the surface with a tiny probe by tunneling electrons.
So the raw data isn't an image at all.
It's just topographical data. You absolutely need powerful computer software to reconstruct that data into a visual 3D map of the atoms.
And beyond just seeing things, computers let us simulate them.
This is a huge part of modern drug design. You use a hybrid approach. For small molecules, you need quantum chemistry to model the electron interactions. For huge proteins, that's computationally impossible, so you use approximations from classical mechanics.
And the Nobel Prize went to the people who figured out how to combine them.
Exactly. Warshall, Karplus, and Levitt showed how you could seamlessly merge both methods. That allows us to accurately simulate how a huge, complex drug might interact with its target before we ever try to make it in a lab.
Then there's the problem of just keeping up with all the research.
The information overload is real. The amount of literature is staggering. So we're now using cognitive computing, things like IBM Watson, that use natural language processing to read and synthesize thousands of papers, looking for connections a human researcher might miss.
Okay, let's pivot to where all this science meets the market, biopharma R&D. The timeline for drug development it's just brutal.
It's a gauntlet, truly. You start with maybe 10,000 compounds to get one single FDA-approved drug 15 years later. And the critical thing to know is that most of them fail, and they fail early.
In the preclinical stage.
Yes, during drug discovery and preclinical testing. This is where you run into the ADME problem: absorption, distribution, metabolism, and excretion.
Let's break that down. What are the big hurdles there?
Well, for absorption, maybe your drug isn't soluble or it gets destroyed by stomach acid. For distribution, the big one is the blood-brain barrier. It's a fortress that stops most molecules from getting to the brain.
Metabolism is the body fighting back.
Pretty much. Your liver tries to break down and deactivate the drug. And finally, excretion. You have to make sure the drug doesn't build up and become toxic. Nanomedicine is trying to solve all of these problems.
And this is pushing us away from one-size-fits-all drugs like ibuprofen and more toward personalized or stratified medicine.
Absolutely. We're now designing drugs for specific patient populations based on their biomarkers. The classic example here is Herceptin.
For breast cancer.
For a specific type of breast cancer. Herceptin is a monoclonal antibody that only works in the 25% of patients whose tumors overexpress a receptor called HER2/neu.
So if you don't have that biomarker, the drug is useless for you.
Completely. Which means Herceptin isn't just a drug, It requires a companion diagnostic test to find the right patients. It's a perfect example of stratified medicine.
This is really the modern version of Ehrlich's old magic bullet idea.
It is. And this is where nanocarriers come in. These targeted drugs can be incredibly powerful, but also highly toxic. We need a way to deliver them only to the cancer cells.
What are some of those delivery systems?
A great example is dendrimers. These are synthetic, star-shaped polymers. Their structure is perfect for encapsulating drugs that don't dissolve well in water, improving their solubility and how they behave in the body.
And what about liposomes? They seem to be everywhere now.
Liposomes are a cornerstone. They're basically tiny artificial vesicles made of a lipid bilayer, just like our own cells. You can load a highly toxic chemotherapy agent, like doxorubicin, inside one.
So you're hiding the poison until it gets to the right place.
That's the idea. The liposome shields the doxorubicin from interacting with healthy tissue, which dramatically reduces devastating side effects like cardiac toxicity. It's a huge win for patient safety.
That's a perfect transition into clinical applications. Let's start with imaging.
Imaging has been transformed. Take MRI. It uses the protons in water in your body's tissues. Nanoparticles, often containing gadolinium, act as contrast agents to make diseased tissue light up.
And the goal is to get even higher resolution.
The research frontier is something called magnetic resonance force microscopy, or MRFM. The dream is to get to nanoscale resolution, to actually image the atomic structure of single large molecules.
You have PT scans, which show function, not just structure.
Right. Positron emission tomography shows metabolic activity. It uses a radioactive tracer, usually a glucose analog called 18F-FDG.
And cancer cells eat up more glucose.
A lot more, so they glow on the PE scan. It's invaluable for finding metastasis or even seeing early signs of Alzheimer's before the brain structure changes. When you fuse it with a CT or MRI, you get function and anatomy in one image. It's incredibly powerful.
Let's move to the most ambitious goal of all, regenerative medicine. This all starts with stem cells. Can you walk us through the hierarchy?
Of course. At the very top, you have totipotent cells, the zygote, right after conception. It can form a complete organism.
The whole thing.
Everything. One step down is pluripotent. These are embryonic stem cells. They can become nearly any cell type in the body, but not a whole new organism. Then you have multipotent, oligopotent, and finally unipotent cells, which are more and more specialized.
And a lot of research is focused on therapeutic cloning.
Yeah.
Why is that approach so important?
Therapeutic cloning, using a technique called SCNT, is about creating patient-matched embryonic stem cells. The huge advantage is that these cells are genetically identical to the patient.
So no immune rejection.
Exactly. No need for a lifetime of immunosuppressant drugs. It's a potential path to treating diseases like diabetes or Alzheimer's by replacing damaged cells with the patient's own healthy new cells.
And for large-scale damage, we're talking about tissue engineering.
Right. This involves using scaffolds, basically biological materials, to guide the regrowth of cells. And the progress is just astounding. The Wake Forest Institute for Regenerative Medicine has successfully engineered and transplanted entire organs like bladders.
That is just incredible.
It is. And we're even finding molecules like the GDF11 protein that have been shown in mice to reverse age-related damage to the heart. The potential for true rejuvenation is on the horizon.
What about the ultimate challenge, restoring sight?
It's one of the hardest problems. When the photoreceptors in the eye are gone, you can't just regrow them yet. So the current approach often involves prosthetic devices.
Bionic eyes.
Essentially, devices like those from Bionic Vision Australia use arrays of electrodes to stimulate the cells that are downstream from the lost photoreceptors. They bypass the damage and send a signal directly toward the brain. Patients can learn to perceive shapes and locate objects. It's a first step, but a monumental one.
We've covered such an incredible range today, from the 10 to the -9 meter scale of a single molecule, all the way up to the massive multi-billion-dollar R&D pipelines that bring these ideas to life. It's clear that the future of medicine is completely dependent on this interdisciplinary approach.
It really is. And for anyone listening, whether you're a student or a professional, understanding this whole ecosystem from the quantum mechanics to the clinical trial strategy is just essential. You can't separate the science from the application anymore.
To help you lock in some of these key ideas, we want to leave you with a quick review exercise.
Personalized medicine relies on biomarkers. So for your review, first, describe the relationship between the drug Herceptin and the HER2 neuroreceptor and explain why this mechanism is a core example of a stratified medicine approach. Then, describe the specific challenge that nanomedicine delivery systems like liposomes are designed to solve when they're used to transport highly toxic drugs such as doxorubicin.